Welcome my dear viewers. This is going to be a very technical topic and believe me, I am trying to make it as simple as possible. Transgenerational epigenetic inheritance. Are we overlooking the future? The future depends on what you do today. So, my dear friends, we have to be very responsible on how experimentation with life and how regulated we are. If you look at this, we talked about test tube babies, and on the other side you see the double helix of the DNA. So, when we are playing with life, we need to know that we have to be very sure is what kind of offsprings are we going to bring into this world. So, 1.5% of all births across the globe today are coming through assisted reproductive technologies. Frightening, isn't it? But nevertheless is true. And why is that happening? It need not happen only for infertility, it also happens because of genetic disorders, it also happens for other reasons because we are now looking at expanding the realms of assisted reproduction not only to infertility but to other reasons as well. So, the question now on all our minds today, are we creating a superior being or are we creating a doom for the future generation? We do not know how safe are our children in future. You may ask me why. Louise Brown, born in 1978, and she has now produced her own child. And why are you talking about this? The FDA has approved it. All countries are practicing it. But my dear children, I would like to tell you that when natural conception takes place, it takes place inside the abdomen, which is very dark. But when you are doing the fertilization, you are doing it under the microscope, under the light. Now, when the gametes are exposed under the bright light, we do not know what is the effect of light on these gametes. Secondly, when you get these gametes fertilized and then they go from day 1 to day 5, they are still under the arc lights. So, what do we really not know is that these babies, though they are born and they produce their offsprings, are there any environmental effects on these gametes and hitherto also the offsprings in future? Time itself will tell us. The front page of the Times said how the first 9 months of your life will shape the rest of your life. The development origin of health and disease hypothesis. This is the perturbation of key events during the periconceptional period and or early development leads to an increased risk of chronic diseases in later life. And this is the Barker hypothesis. Now, we will have a look at this. These are all the babies that have been attributed to assisted reproductive technologies. I know this is quite frightening to all of you. Beckman-Weidman syndrome, Silver-Russell syndrome and oculo-auricular vertebral spectrum. I am sure all of you have been told that IVF is a very safe procedure. Of course, it is still a safe procedure. I am not trying to be an alarmist, but nevertheless at the back of your mind just keep this in your mind that it also has its drawbacks, and of course ART and disorders since 2002. Well, what are the important questions that we have to ask ourselves? Is ART associated with an increased risk of serious disorders? If the associations are genuine, what are the causes? What are the transgenerational effects of these? So, the term epigenetics, which was coined by Conrad in 1942, explains that how environment plays an important role in shaping the genetics of the future generation. So, the study of mitotically and meiotically heritable changes in gene function that cannot be explained by changes in the DNA sequences. So, gene marking and gene regulatory system essential for normal mammalian development. So, the genetic vision, which is the 46 chromosomes, and the epigenetic vision is different susceptibility to early or late onset disease. And why does that happen? The genetic vision would be the normal genetic vision, but when you look at the epigenetic vision is when these embryos are outside the body and then get implanted into the uterus. So, what is this environment influence on these babies? And only as we go through transgenerational research we will be able to find out. So, the epigenetic regulations would be at the DNA methylation, histone modifications and RNA transcripts. So, life cycle of an epigenetic mark will be erasure of epigenetic marks from primordial germ cells or establishment of new set of marks during gametogenesis, genome wide erasure of methylation during the pre-implantation stages, de novo establishment of marks during development and differentiation from around the blastocyst stage, and there is what you will see, a pictorial depiction of what I have just said in my previous slide. So, assisted reproduction procedures are performed at a time when it is dynamic. Essential epigenetic reprogramming events are occurring in the gametes and embryos. Yet, the extent of these epigenetic changes and the relevance to the human health and disease in assisted reproductive cohorts is only just beginning to be understood. So, if you were to look at this graph, the epigenetic genome reprogramming during gamete and early embryo development. So, you will see in this slide that at the DNA methylation stage, at the superovulation stage, the inner cell mass, the ICSI and the IVF, which is an artificial fertilization technique, and also the embryo culture, which is different from what you will have in the follicular fluid and also in the tubular fluid. So, the embryo biopsy, the cryopreservation, these are all environmental factors which are going to play a very important role in modifying the kind of environment that these embryos are going to be exposed to compared to a normal conception. So, this is the same that we are going to have, and I will just explain this slide a little better. This may look like a chakra view, all right. The hormonal ovarian stimulation in vitro cultures, that has an effect, that can also have an epigenetic variance. Secondly, it can cause a risk of major malformations, abnormal implantation, abnormal placentation, risks of placental insufficiency and risks of preeclampsia, risk of premature birth and a risk of cesarean section. Oh my dear, I didn't realize that all this came from assisted reproductive technology, but my dear, the epigenetic role has only just begun to be understood. So, risk of intrauterine growth retardation and the risk of perinatal death. So, therefore, the role of genetic variance, epigenetic variance and exogenous factors in adverse health outcomes in assisted reproductive technology pregnancies and children. So, therefore, one has to be clear that we are not without danger, and we have to be clear in ourselves that let us just go ahead and be careful when we do IVF left, right and center without some meaningful indications. Disorders of genomic imprinting and human assisted reproduction means genomic imprinting is defined as the exclusive or predominant expression of one allele of a gene from either of the maternal or paternal allele. The monoallelic expression is regulated by allele specific epigenetic marks such as DNA methylation. Current hypothesis, genomic imprintation may allow the exertion of parental epigenetic influences on the growth and development of the conceptus. So, in Human Reproduction in 2015 shows a systematic review and meta-analysis of DNA methylation levels and imprinting disorders in children conceived by IVF-ICSI compared with children conceived spontaneously. So, there is an association between ART and imprinting syndromes, but no evidence of generalized changes in DNA methylation of selected genes. So, if you were to look at Beckworth-Weidman syndrome or Silver-Russell syndrome, you will see that with the first one, you will have macroglossia, that is a big tongue, an umbilical hernia and an omphalocele. With the Silver-Russell syndrome, you will see the problem lies with the chromosome 7 and 11, and you will see this child looking different from normal children. Now, are imprinting disorders more prevalent after human in vitro fertilization or intracytoplasmic sperm injections? There have been 8 studies. The relative risk of 5.2 is very important, that the confidence interval of 1.6 to 7.4 indicating that one Beckman-Weidman syndrome child will be born for every 2,700 IVF-ICSI births when using a population prevalence in the general population of 1:13,700. So, what is the effect of in vitro culture? Now, in vitro culture effects, it can be at a cellular level, a development level, a metabolic level. So, the differences in gene expression profiles between human pre-implantation embryo cultured in different IVF cultured media has been actually shown in human reproduction, and we will look at what it could be, the effect of in vitro culture on the birth weight. This has shown large offspring syndrome in cattle and sheep, which means that bovine and ovine embryos exposed to a variety of unusual environments prior to the blastocyst stage have resulted in the development of unusually large offsprings, which can also exhibit a number of organ defects. Now, characterization of global loss of imprinting in fetal overgrowth syndrome induced by assisted reproduction. So, the question we have to ask ourselves, does the type of culture medium influence birth weight of children born after IVF? Well, factors affecting birth weight would be the age of the media, length of the culture and the protein source. Time to take human embryo culture seriously. The complete formulation of culture media to be disclosed, and all changes should be justified, validated and communicated to end users by the manufacturers. Changes should be made to the existing regulatory system to achieve transparency and improve monitoring of outcomes to the long-term benefit of ART children and society. The effect of superovulation on epigenetics overrides the progressive oocyte growth dependent process of epigenetic maturation and imprint establishment, lead to the recruitment of poor quality oocytes that would not normally be selected to ovulate. Perturbed genomic imprinting of maternally and paternally expressed genes in the embryo and placenta disrupt key oocyte early embryo specific factors important for imprint maintenance during pre-implantation and development. So, superovulation in mice alters the methylation pattern of impaired genes in the sperms of the offspring. Aberrant DNA methylation of imprinted loci in the superovulated oocytes, and superovulation influences methylation, reprogramming and delays onset of DNA replication in both pronuclei and mouse zygotes. Epigenetic changes in human assisted reproductive cohorts. Are there subtle genome-wide epigenetic alterations in normal offsprings conceived by assisted reproductive technologies? these data suggest is that ART is associated with widespread epigenetic modifications in phenotypically normal children, and that these modifications may increase the risk of adverse cardiometabolic outcomes. Adult cardiovascular and metabolic diseases, a possible legacy of assisted reproduction. Systemic and pulmonary vascular dysfunction, right ventricular dysfunction, cardiovascular remodeling in utero, and altered glucose metabolism. So, these are all the various areas in which epigenetics plays a major role. So, right ventricular dysfunction in children and adolescents conceived by assisted reproductive technologies, and the original article suggests that assisted reproductive technologies are associated with CVS remodeling in utero that persists postnatally. The longer term health outcomes for children born as a result of IVF treatment, part 2 is the mental health and developmental outcomes. It is possible that these outcomes as a result of the alteration, adaptation of metabolic pathways in mammalian embryos exposed in suboptimal culture media and or environments. Many enzymes involved in epigenetic gene regulation in eukaryotic cells make use of co-substrates and cofactors generated by cellular metabolism, like B vitamins and the elements such as sulfur, zinc and cobalt. These in turn are influenced by lifestyle factors including obesity, cigarette smoking, alcohol and caffeine consumption. So, there is a light at the end of the tunnel. And are we looking at assisted reproductive technologies that are associated with limited genetic variations at birth that are largely resolved by adulthood? So, this is 2019 Nature's Review. So, what are the things that we can look at in a very serious way that we can modify? Certainly, we can modify maternal unhealthy diet, the stress, the smoke, infection and obesity, the epigenetic changes of the altered programming growth and metabolism, and increased risk of adult onset disease can be minimized. What are the environmental stresses? High fat and high sugar content, obesity, stress, traumatic experience, low level of exercise training, alcohol and cigarette abuse, and toxic environmental agents. These epigenetic changes can have its effect on both the oocytes and the sperms, and the maternal impact as well as the paternal impact at that time during the periconceptional period and at the time of fertilization lands up at the time of the female pronucleus and the male pronucleus. What then happens after fertilization is that during embryo development, the altered embryo development, the phenotype and long life sequences. We know that environmental factors do affect, part of which we can actually change, like things, lifestyle modifications, things that are we take for granted, but there are things that we cannot change. Like for example, where the egg and the sperm is taken outside the body and then fertilized. So, we can actually look at what are the culture medias and whether we can make it as natural as possible, and also for the fact look at the pregnancies properly. We can't do much about the genetic changes, but nevertheless whatever we can do, we can limit the epigenetic changes. The only thing that we cannot do is we have to be vigilant that all is not hunky dory. We do have epigenetic changes. So, IVF cannot be taken very frivolously. We have to look at it, and the indications have to be regulated, so that when there is a need for IVF, we know that we have to take the risks and benefits, because epigenetic diseases have to be looked at very seriously. So, my friends, we do not fear the unknown. We fear what we think we know about the unknown. Thank you very much.